Assalamualaikum.
Walaikum assalam wa rahmatullahi wa barakatuh. Good morning to you, all of you boys. I hope everybody's doing well, inshallah. Everybody's good. So let's start for today's topic. Today's topic is about testing the leaves for the starch. We will be doing a small test. We will be looking at a video, a demonstration about testing. Yes, Wahib, hello to you. Testing that if the plants, the leaves, they are making the glucose after the photosynthesis, will they be able to store that glucose in the form of starch? So let's have a look at this. We'll start the lesson with this video. This will, I didn't copy it. Okay, let's copy this. All right, let me turn on the voice.
Glucose through photosynthesis. The excess glucose is
Let's begin from the start. So we're going to test. Those who just are coming in, we're going to do a small test on the leaves and see if they contain the starch. The first experiment that we'll do, I'll keep the voice closed, I'll explain along with it, is about to see that the leaves of the plants, they produce glucose. And once they have done this, the excess amount of glucose is going to be converted into starch, which is actually a stored food, which is a reserved food material for the plants. Because sometimes there will be days when there is not enough sunshine, so the plant will not be able to do photosynthesis, or at the night time when they are repairing their bodies, they need extra glucose to grow and to repair themselves. So what do? They actually eat back the stored reserved food in the form of starch. So let's see. In this experiment, let me explain a few things.
we need a leaf, boiling
You need the leaf, some water where you can boil. This is ethanol, and then this is iodine solution. A stand with a Bunsen burner, so you can boil the water, right? A test tube and a Bunsen burner. So he's flaming up the Bunsen burner, and he's gonna boil the water. Once the water is boiled, we will insert the leaf, or you can insert the leaf before it boils. So after like 30 to 40 seconds, he has taken the leaf out and he's putting it in a test tube. The test tube, and then he would add ethanol to it. This is the ethanol which is added. Why are we adding ethanol? So we can actually extract the chlorophyll out of this. If it is properly extracted out, the leaf will become white. In this experiment, you will not be able to see it, but the second experiment, I have another one where we can see this is actually becomes absolutely white. And this is the dangerous part. We should not keep the flame running if we're going to insert the ethanol, because ethanol is actually uh, very flammable. Now, if he would have kept this for 2 to 3 minutes, this leaf would have become absolutely white. It is still green now, but if it stayed for 2 to 3 more minutes, it would have become absolutely white. Now he is dropping iodine, iodine solution to check the starch. If the starch is present, the leaf will become dark blackish, bluish, or brown blackish. That is to confirm that yes, the starch is present. So this was the half of the experiment. The other half is, what if we have a leaf which didn't do photosynthesis for about a long time, for 5 days or 3 days? So I'll show you a video of this as well. this is a video where we will see that there's a leaf with one doing photosynthesis and one leaf which was deprived of sunlight. It didn't get any of the sunlight and it wasn't able to do photosynthesis for 3 to 4 days. And what happens to it?
Photosynthesis is the natural process occurring in green plants that converts solar energy to chemical energy and releases oxygen into the atmosphere. Starch test is conducted to prove that sunlight is necessary for photosynthesis. Initially, de-starching of some leaves is done. Some leaves of the plant to be studied are covered with a strip of paper or foil. This is done before dawn to block the sunlight. Any starch present in the plant leaves after de-starching is formed under experimental conditions. The plant is then exposed to light for few days. Few uncovered, exposed leaves and leaves that were covered with foil are then detached from the plant. Starch test is then performed on the detached leaves to examine the presence of starch. Detached leaf from the plant is dipped in boiling water for 30 seconds. Boiling the leaf in water destroys the enzymes, thus prevents all the further chemical changes during the experiment. Leaf is then put in ethyl alcohol to dissolve the chlorophyll. Leaf gets decolorized due to the loss of chlorophyll pigment and becomes pale yellow in color. The decolorized leaf is then washed in water and dipped into the dilute iodine solution. Presence of starch can be detected visually by change in the leaf color. Leaf exposed to sunlight turns blue-black or bluish.
Now this is the result of the leaf, which was a normal leaf, which was allowed to do photosynthesis for normal as usual. But now we're going to go to the second leaf, which was deprived of sunlight for 3 days. We covered it up with aluminum foil, which blocks the sunlight 100%. Let's have a look at it.
Purple in color, which demonstrates that the photosynthesis was going on in the leaf, therefore producing starch. On the other hand, leaf that was covered does not show any change in the color much evidently, and hence gives brown color.
Now this is the leaf which was covered in the foil for 3 to 4 days, and it wasn't able to do any photosynthesis. So when it was deprived of making its own food, it was eating all the starch that was present in the leaf. And now after 4 days, they have also run out of starch. There is no starch available in this leaf. This test proves that the sunlight is needed to do photosynthesis, and the photosynthesis will make glucose, and the extra glucose will be stored as starch.
With iodine, since the covered leaf did not receive the sunlight, no starch is formed in it.
So this is the normal one. This is with the starch. This is without starch. This was doing photosynthesis. This was not doing any photosynthesis.
So, it is inferred that plant manufactures starch in the presence of sunlight by the process of photosynthesis.
Alright, so let's have a look at another video where we are going to do the same test but with another person, so you might be able to see the difference.
Hi, today what we're doing is we're testing a leaf for starch. We're testing a variegated leaf, and that is one which has chlorophyll in some bits and not in other bits. I'll just put it there so you can see. In order to do this, we're going to need some hot water, we're going to need a tripod heating equipment, and we're going to need some iodine solution and some ethanol and some safety specs. We'll don the safety specs now. Right, let's get this water fully boiling. Lighting on safety flame, putting it onto full roaring blue flame, and let's get that heated up. Now what we're going to do is we're going to boil this leaf from this poor draggled plant here, which maybe has seen better days. We're going to boil this in here for about 20 seconds or so, and what that will do is it's going to denature the enzyme, it's going to kill the leaf, and that will stop photosynthesis. What we're trying to do is see where the leaf is now. So we're going to stop all the living processes going on inside this leaf by denaturing its enzymes.
This point is very important, that why do we put the leaf into boiling water? The answer is, number one, to kill all the living activities inside the cell, to kill all the cells. Number two, to stop all the chemical reactions happening, to denature the enzyme so that none of the activities in the enzymes can be done inside the cell. All right?
Great, our water is boiling now. I'm going to turn that off here. It's hot enough, I don't need to keep it boiling. I'm just going to get the leaf under the water. There we go. And we're just counting for about 20 seconds. That's all that's needed. It's going to soften the leaf, it's going to soften the cellulose fibers in there as well as denaturing the enzymes. So I think that will do. Now importantly, we've switched off our flame, because now what we want to do is submerge our leaf in boiling ethanol. Ethanol, of course, is extremely flammable, and so we don't want any naked flames near it. And we'll boil the ethanol by putting it into our water bath here. So out you come, Mr. Variegated leaf, pop you in there, and we're going to leave that in there boiling for about 2 minutes. What this is going to do is the chlorophyll doesn't dissolve in water. You know that when you boil broccoli at home, you don't get white broccoli at the end of it because the chlorophyll stays in there. It's not soluble in water. It is soluble in ethanol. And we are now going to remove the chlorophyll. That's not because we're expecting photosynthesis to carry on. It's because we're going to be doing a color test on this at the end of it, and we don't want any green pigments from the chlorophyll in place to interfere with our test. Well, our ethanol is boiling very nicely now. I'm just going to hold this under our camera here. You can see it boiling really, really well. We're going to leave that for 2 minutes and come back to it. Yeah, that is pretty much there. It's done very well. We've done our 2 minutes, we've dissolved the chlorophyll, and I'm going to hold it under the camera here so that you can see that. It's got a little bit hot in the meanwhile, so I'm going to use my tongs. And you can see there, we've dissolved the chlorophyll. It's gone off into the ethanol, and you can already see the leaf is white underneath it. Okay, we're going to pour off the ethanol. We're going to pour that actually straight back into here. And also we're going to put the leaf into there as well. The leaf, well, I'll show you first before I pour it in. It's going to come out all kind of crispy. It's rather peculiar. Out it comes. It's all kind of crispy. It's not floppy at all. Indeed, if I can probably make you hear it. Well, I'm not sure. Here we go. So we're going to drop it into here. That'll soften the leaf up. Because we need it soft, we're going to need to spread it out onto our white tile in order to look for our results. There we go. We'll leave that in there. So, on to our leaf down here. Now, we'll remind ourselves why we're doing this. We're looking to see where the starch is in this leaf. That's because the starch is indicative of photosynthesis having happened. Photosynthesis makes glucose. Glucose is polymerized and stored as starch. So the regions of the leaf where there is starch, that suggests that that's where photosynthesis has been occurring. Okay, let's go. and we're going to put our iodine solution in it. And we're going to drizzle our iodine solution onto it. There we go. We'll let it we'll let the stain take for just a few seconds, and that's very beautiful. We can see that really nicely. I'll just zoom into it. Here we go. And you can see here the pattern of the starch, that's the black areas rather than the unstained orange areas. The black areas are where the starch is.
What he's trying to emphasize on, the black region is the region where the starch is, and the orange one is where there is no starch. So it shows that this is the region. Yes, please grow up. Come on. So this is the region where we have to focus on. This is the region where the starch is being, safe, and the orange one is the one where it is not available. So starch is not thoroughly distributed. It is in some part of the leaf, not completely covering the whole thing. The borders, which are the orange one, do not contain the starch.
And also they correspond exactly to where the chlorophyll was. And therefore this is evidence, it's not proof, but it's evidence that the chlorophyll is required to make glucose, to make starch. The chlorophyll is required for photosynthesis. Thank you.
All right, that's it. Let me show you another one. And that would be the last. Yeah, that was the last one. So we have to go to the kahoot.it. You have to start kahoot.it. I'll give you the code. So let me send you the ID, the pin through the chat. If you have the app, please use the app, or you can go to the browser. It's kahoot.it, and the code is 390-3105. 390-3105. So please log on to the Kahoot. We have a small, you don't have to like it cut. Keep your names appropriate so that I can keep a record of you guys. Yes, Mohammed is here, Ryan is here, good. No laser beams, sorry. Okay, let's keep the I like it cut and I'll see who is missing. I can trace you back with your IP address and then I'll be talking to your parents. This is your last warning before I go and get you. You remove your cut G yourself. The other one as well. Remember, your IPs are with me through the canvas, so it is easily traceable. So we have Alan, we have Yas, Walid, and we are left with 15 minutes. So please, yes, Yad, I have done my master's in IT. I'm a software engineer as well. So let's go to the start. 69 guy, 420, you're on my hit list now. I'll be talking to your parent very soon. This is getting a recorded session and you have been warned. So let's begin. We will be looking at the photosynthesis, and this is the quiz. Why do we need to boil the leaf to test for the starch? Why did we boil the leaf in the beginning? So to get rid of the chlorophyll, to kill the leaf, to stop any chemical reactions, or just because that's not a reason. So you have to choose between red, yellow, and blue. So good job, 6 boys have done it right. It is to stop any chemical reactions, even if you chose to kill the leaf, that is acceptable. That is the correct answer. To get rid of the chlorophyll, we will use the ethanol. So Muhammad is on top, Badr, Iyas, Waleed, and Riyad. Good job, boys. We have, why is ethanol used? To denature the bacteria, we didn't discuss bacteria, to see if the starch is present, to add the color, to get rid of the chlorophyll. So remember when we were adding the ethanol into the leaf and there was a green water coming out? It pretty much explains this part that what were we doing? Last 15 seconds. Why do we use ethanol? To add the color, to see if the starch is present, to get rid of chlorophyll, to denature bacteria. And the answer is to get rid of the chlorophyll. The green color that is coming out is to get rid of the chlorophyll. So that's why the leaf was becoming pale whitish. That's why because we got rid of all the chlorophyll. So ethanol removes the chlorophyll. So we have a new person on top, which is Badr. Very good. Waleed. Muhammad is okay. Muhammad is now number 4. So let's see. Why is iodine solution used? Why did we put the iodine solution on the leaf? To see if the starch is present, to kill the leaf, to remove the chlorophyll, to denature bacteria. So it's a simple question now because the options are very easy. We have done to kill the leaf, we have done to remove the chlorophyll, and bacteria was never in the picture. So you know what's the answer. All right, good job. 5 geniuses. And they said yes to see if the starch is present. Good job. Now, who's on top? MashaAllah, Badr is still on top. He's on fire. Muhammad Abdul Dail, you are still close. Now, let's go to the next question. If the starch is present, what color will the leaf turn to? Choose the options below. Is it going to be dark brown? Is it going to be white, purple, purple blue, or blue black? I'm in the middle of class. So the blue-black color is the right answer. Even if somebody has chosen purple-blue, that would have been right. I would have accepted this right. So good job. Let's see the scores. Oh, there's a big upset. Ahmed is again on top. Very good. Yes, in second position. Waleed is on third. So come on, boys. Maintain your position. Hatib is here. Those who are logging in, they can see the pin code below. Join on the website. So the question is, to test for the chlorophyll, what type of leaf should be used? What type of leaf should be used? Um, I'll give you the options to choose between the yellow and the green. The one which says non-variegated and variegated, that means one with the vessels inside, the tubes, the xylem and the phloem is in there or not. So which one of the leaf are we going to use? Choose between yellow and green. Square or the circle option. The square is the right option. And the people who chose blue, I'm telling you not to choose blue. Choose between yellow and green. So variegated. And we have Waleed on top. So it's a big upset. Good job, Waleed. For chlorophyll test, after adding iodine solution, what color does it? This one we didn't take, so we'll skip this question. We'll go to the next one. If a leaf contains chlorophyll, it is able to do photosynthesis and produce starch. Is this true or is this false? Is this true or is this false? This is the easiest question. Good job, everybody did it right. Excellent. So, yes is on position 2. Very good. Why do you need soda lime for carbon dioxide experiment? We didn't do this, so we'll skip it. Let's go to the next question. Ninth question. What's the equation for photosynthesis? How do we write photosynthesis? Is it glucose plus oxygen equals to carbon dioxide and water? Second, glucose is equal to ethanol plus carbon dioxide. Number 3, carbon dioxide plus water equals to glucose. Or number 4, carbon dioxide plus water equals to glucose plus oxygen. It's a very simple question, but you have to be very careful when you're choosing the answer. Answer can be a tricky one. Photosynthesis. 2 people got it right. See, I was telling you about the trick part. The carbon dioxide and the water equals to glucose plus oxygen. You have to mention the oxygen. Let's go to the last question. Glucose is stored as in the plants. Is it stored as sugars, sucrose, starch, or glycogen? Two boys left. One answer left. It is stored as starch. 2 people were able to answer it right, and it can be stored as sucrose. So if somebody have chosen to use sucrose, you would have been right, or starch. Glycogen is in the animals. Glycogen is in the animals. And sugar is not a storage. Sugar is the basic molecule like glucose. So let's see the results. Number 3, yes, good job. Number 2, Walid. Good, Walid. And number 1 is Muhammad Abu Judail. Excellent job. Very good. And runners up, Muhammad and Badr. So very good. So let's see the difficult questions. We got 44% people as a team. We had 44 people doing it right. 5 questions were difficult for you all. I would like to see if anybody would like to read these questions with the right answer. Who would like to do this? Okay, so nobody's raising a hand. The first one was to test the chlorophyll. What type of leaf should we use? It should be variegated. Equation should be with carbon dioxide and water, resulting with glucose and oxygen. The glucose is stored as sucrose or starch, these 2. Why was ethanol used? To get rid of the green color, the chlorophyll. And if the starch is present, what color will leaf turn to? It will turn into blue-black. So that would be all. And let's view the full report. We're done with the session for today. I'll stop the recording.